Welcome everyone to the Diatom Web Academy. This is being brought to you by the Diatom Taxonomic Certification Committee, and we're a group of people that have been working through the Society for Freshwater Science to offer a certification program on diatom identification. And we ended up in the course of developing that, we found out that we needed to do a little training. And so this Web Academy is an offshoot, and we are offering this series of presentations to support training for analysts, taxonomists, managers, academics, and students to further consistency in diatom data and analysis. So you can access previous presentations through the Diatoms of North America. I'll post that shortly. And this presentation will be recorded and accessed soon thereafter. Our next presentation will be in 2 weeks on Tuesday. Vicky Chairabi and Shelley Wu will be presenting a seminar on golden brown diatom opportunities in education. So I will turn this over to Sylvia to introduce our speaker for today.
Thanks, Sarah. It is my pleasure to introduce Dr. Paula Fury. She's professor in the Department of Biology at St. Catherine University in St. Paul, Minnesota. Paula is a freshwater ecologist, and her research has quite a global scope, from the wet walls of the Great Smoky Mountains and the Eel River in California to the Peruvian Andes, as well as Iceland, just to name a few places in the world. Paula also joined the Diatoms of North America team as a member of the editorial review board, and she is the content editor for the Eunotia Diatoms. So welcome, Paula. The floor is now yours.
Great. Thank you. I'm super excited to be here. I like talking about Eunotia, and I hope you enjoy the content from today. As an educator, I like to put people to work when I present. So I hope you saw that little note up at the top right-hand corner to get a pencil and a piece of paper ready. We are going to do a little bit of prep work on your end so that you know what to listen for while I am presenting. So I would like you to get a pencil and a piece of paper, number it from 1 to 10, flex your fingers, and I'm going to do a little preparation quiz. We're going to go through the quiz fairly quickly, so don't worry about whether you know an answer. You can make something up, you can make a sketch, and then you'll get the answers as we present. So, are you ready? Here we go. Question number 1. What is the significance of the number 1837? So 1837. Question number 2. Where is the Raphe located on Eunotia? Again, you can make a sketch, capture your response any way you would like. Question number 3. How many Rimoportulae do Eunotia have? How many Rimoportulae? Question number 4. Are Eunotia wider in valve or girdle view? Valve or girdle view, which is widest? Question number 5. What is the significance of the number 7.5? So 7.5. Question 6. What is the significance of the number 337.5? So question 5 and 6, we've got 7.5, and 337.5. Question number 7. If you had to find a Eunotia in a diatom scavenger hunt, where would you go? Bonus points here when you're grading your response for alliteration or rhyming. So where would you go to find Eunotia in a diatom scavenger hunt? Question number 8. Do Eunotia form stalks? Do Eunotia form stalks? Question number 9. What kind of dance moves do Eunotia have? What kind of dance moves do Eunotia have? And then question number 10, it's a little bit of a challenge question. I'll buy you a little bit of time to fill in responses you may have missed earlier. But name 3 words associated with Eunotia morphology that begin with the letter R. And if you're really fast on that, try S or try H. So 3 words associated with Eunotia morphology that begin with the letter R, or S or H if you get through the R. So those are my prep questions. I hope you had a little bit of fun with them. You'll be able to grade your responses as we go along. It's all for fun. I'm not collecting grades at the end. But the reason that I wanted to ask you about those questions is that the Eunotia is just a cool group, but it's really challenging. So for my presentation today, I'm going to talk through the characteristics of Eunotia, and embedded in that, bring forth some of the identification challenges and get you thinking about if you're new to diatoms or not new to diatoms, new to Eunotia, not new to Eunotia. When you're looking under the microscope, what sorts of things do you need to train your eye to see? So we're going to kind of up the Eunotia goggles that you're wearing and get you looking for some key characteristics. Or if you're describing a Eunotioid taxon, what might you want to pay attention to, kind of above and beyond some of the typical diatom things. As we're working through the typical characteristics of Eunotia, you'll get a little bit of a tour of the diversity, and you can also use those Eunotia goggles as we're going through the diversity. And then we'll talk a little bit about the ecology and kind of get you thinking about that morphology of Eunotia and what it means if you were going to be a Eunotia out in whatever environment that you are in. And then of course we'll have some time for some Q&A. So as a group of diatoms, there's lots of diversity within Eunotia. They have a global distribution. They're one of the groups of diatoms that has high diversity within it. However, within all of that diversity of Eunotia, there's also high variability in valve morphology, the location and shape of the Raphe, the number and location of the Rimoportulae, different growth forms. All of this makes Eunotia challenging to identify, especially if you're doing a count and you're looking at kind of one valve at a time. It also makes it a little bit challenging to pull out what we would call like stable characteristics that don't vary within a species or are consistent. So we're going to talk through those pieces and hopefully kind of increase your confidence for looking at Eunotia. So here we go. Answer to question number 1, 1837, right? That's when Eunotia was first described by Ehrenberg, the type species being Eunotia arcus. One of the things that, kind of if you were looking at a microscope and you saw something that you thought was Eunotia, you would see that it was dorsal ventrally flattened, right? There's a convex dorsal margin, and the degree of curve of that dorsal margin can change. The ventral margin can also be kind of concave to straight. Yeah, and asymmetrical about the apical axis. But one of the things, there was an earlier presentation that was made about how to define a species, and the phrase came up really thinking about a species of diatom as a size and shape series. And I think that Eunotia really fit that bill of you really have to capture Eunotia diversity across its size range, and you have to pay attention to the shape. So there's 3 different examples up on the screen, but you can see, for example, in Eunotia Ehrenberg that the curvature of the dorsal margin is much more pronounced in the smaller specimens than the larger ones, those types of things. So thinking about the size and shape series. One of the challenges, again, with identification is that not only do Eunotia species vary between species, but within a species, that shape, those different shape characteristics that you're looking at, right, can vary very a lot. So if you happen to look at a long or large valve and a small valve and you saw them side by side, unless you had captured that size series, you might not connect the two together as belonging to the same species, right? Maybe it's the dorsal undulations, if they are present, are more pronounced in one size over another. So just something for you to think about and always kind of be tapped into. If somebody is sending me Eunotia specimens to identify, I always ask for more than one image of the specimens that you're looking at. That helps to kind of figure out that variability. So variation in size. Here's another example, right? Eunotia papilliforma, right, where one end is wider, one end is narrower than the other. But how that plays out in terms of the overall valve morphology changes or is quite variable, right? Maybe in response to environmental characteristics or just that genus has a little bit of a variable shape. And you need to be able to see and recognize that to separate it from a taxon that has very similar morphology. So taking very careful length and width measurements and capturing that size series can help with that. So it's one of the challenges for identification, but it's also one of the really cool things about Eunotia, I think. All right, answer to questions 5 and 6. So we have the Diatoms of North America website, which is populated with some of the Eunotia species. There's lots of room to do some work there. But 7.5 is the shortest valve that is captured on the DONA website, and 337.5 is the longest one that has been recorded. That doesn't necessarily mean that 7.5 is the shortest Eunotia or 337.5 is the longest Eunotia. I think there are some taxa that kind of can go outside those ranges, but on the DONA website, this is the shortest and the longest Eunotia, which is just crazy. That's a pretty wide range in valve length. Striae density can also vary, right? And these are, you know, kind of typical diatom characteristics that you might look at when thinking about diatoms in general. But certainly for the Eunotia, it can help you capture what species is what species or how it varies, right? So the striae can be particularly dense, or they can be particularly sparse, right? Those striae characteristics certainly apply here. So let's think about the striae for a second in Eunotia, right? They're typically uniseriate. They are, the areolae are small and rounded. And when you're looking under the microscope, you can pay attention to kind of the robustness of that. Are the areolae easy to see, right? Or are they particularly faint and you really have to kind of have the resolution on your microscope set up clearly to be able to capture them? And again, here, there's lots of variability in the striae in Eunotia. They can be have full extension across the valve face. There can also be short striae up along the dorsal margin. Those can appear in some taxa. There are also some Eunotia species where the striae don't extend across the valve space. Most of the ones that we have on the DONA website right now have the striae going all the way across the valve face. There's a blue arrow pointing to a hyaline area. You can think of it as the sternum. That shows up in some Eunotia species, and you can pay attention to that or other hyaline areas that appear. Certainly would be helpful. And you'll also see on the screen, there's a couple arrows pointing to Raphe and the Raphe fissure. We're going to talk about that in fair detail, but pay attention to the striae. So this is the external expression of the striae in the areolae under light microscope and scanning electron microscope. But something that people don't necessarily think about or capture in their descriptions of Eunotia is the internal expression of the areolae and kind of what's happening in different species. So internally, the areolae can be flat or kind of flush, or they can be sunken into a little groove. And I think there are some patterns there across species and species complexes. So if you can capture that, that might be really helpful in terms of our overall understanding of patterns within Eunotia. And you can pay attention to, are the striae parallel, or do they start to radiate at the ends? Sometimes the striae density at the ends of Eunotia get more dense, so you can measure the density of the striae in 10 microns in the center of the valve, and it's also helpful to do that at the apices of the valve. But an internal expression of areolae can be something that wasn't on your list of things to pay attention to. See if you can capture it in the future. The Raphe, probably one of the most interesting characteristics for Eunotia, and one of the reasons when you think about diatoms as a whole, why people pay attention to the Raphe and its characteristics, right? From a systematic point of view, how do Eunotia fit into the development of Raphe, the Raphe system in diatoms as a whole? But if you are looking at Eunotia, there's lots of characteristics around the Raphe that you need to pay attention to, particularly with respect to location and subtleties around the shape. So Eunotia have Raphe that is present on both valves. It is typically relatively short and can be often quite short. It occurs kind of at the valve apices, varies in length, doesn't have a central nodule. But the most interesting characteristic is that the Raphe is on the valve mantle, right? If you think about a navicula or naviculoid type diatom where the Raphe is in the center of the valve face, in Eunotia, they start out on the valve mantle. This Raphe can then extend up onto the valve face, and then there can even be a further extension of a Raphe fissure that can recurve back quite strongly. It can zigzag a little bit. So the upper left image where there's Raphe and Raphe fissure marked on the screen, right, is a good example of a strongly recurved Raphe system, right? Or in the bottom right-hand side of your screen, you can see that Raphe fissure end going all the way up to the dorsal margin. Or the Eunotia incisa in the middle of your screen at the bottom, right, where the Raphe is remaining on the mantle. How cool is that? So if this was question 2 in your quiz response, you can decide how much credit you give yourself, right? Did you capture this variability in your response, or did you just capture part of it? Maybe you only give yourself 0.5 out of 1. I'll let you decide. And I want to pause here for a second because I want you to think about, well, why is there all this variability in Raphe location and shape, and what does it mean if you're in a Eunotia with one type of Raphe system versus another, and how you then interact with the environment? That's an interesting thing, I think, to think about. So Raphe location and shape. There's a fairly recent manuscript, and I think actually the original version of this that came out in 2009, that is proposing some terminology for describing the Raphe system in Eunotia. And regardless of whether you agree with kind of the proposed terminology here, the point that I want to put out to you is that when you are looking at or describing the Raphe of different Eunotia species, there are lots of features to pay attention to, right? So if the Raphe is located on the apices, right, how does it show up on the in girdle view? How does it extend up? You can take a look at the so on the right hand part of the screen there, top 2 images, there's a Raphe system without terminal fissures, and you can see some variation in how it kind of shows up on the valve face, whether there's a pore or whether it's kind of like a funnel-like ending to the Raphe system, right? And you can also be very mindful of the location, right? Is it in the middle of the valve pole, or what you could call the ventral corner of the valve, right? Or does it extend all the way up to the dorsal margin? Same thing if you're so in the bottom 2 pictures there, if you're thinking about the Raphe fissures, right? How does it end? Does it end in a little, kind of a little widening, what in this paper they're calling a little lacuna ending? Or is it just kind of end? And how does that kind of fissure extend? So Raphe system and Eunotia are super interesting. So you notice at the top of the screen, there's a series of terms that are listed. So mirror symmetric, right? So what you see on one side is similar to the other. It's on the mantle, so you can think of it as a mantle offset Raphe system, and then brevaslit, right? So shortened to the poles of the of the valve. So if you have the opportunity to look at a scanning electron microscope or have really good light microscope images, you can clearly define what that looks like. So here's some examples of a terminal fissure and a little bit of a lacuna end to that fissure. And then sometimes even internally, right, you can tell where the fissure is because there would be a hyaline area. That's the area circled in red there on that Eunotia pseudoflexa. So it tells you a lot about the Raphe system and kind of how it's unfolding, which I think is super cool. And I think to varying degrees, we've done a good job or not a good job of describing some of these features. And I think there's some patterns here in the Raphe system that might help us understand Eunotia and Eunotia complexes a little better. Linked with that, you can think about the halictoglossa, right? So that kind of the ending of the Raphe there. And when you're looking under the light microscope, you often can see it. That can be helpful in diagnostic. Sometimes it's a little bit less prominent, but certainly helpful to look at. And internally, when you look at the halictoglossa, right, where does it show up? So its location can be helpful in understanding what you then see externally for the Raphe system. I like thinking of taxa like Eunotia incisa, where the halictoglossa is particularly inset from the apices, and it gives the Eunotia kind of a nose-like ending. That can be a characteristic that can help you identify species or potential set of species. Or something like Eunotia papilliforma or rhomboidea, where the inset can appear a little bit varied on each end. As you can see, the dorsal margin's doing something a little bit different. But you got one end that looks nose-like and one end that doesn't. So that terminal nodule is something that is worth paying attention to. Here's an example from the DONA website, Eunotia lewisii, which if you go to the website is one of the first images that pops up today. I don't know if that has been there a while or if it was just for this week of Eunotia presenting. But here's an example where you can see the halictoglossa, and there's also that kind of an extension of silica going up to the top. That's a little bit unusual and is helpful to identify this particular species. You'll also notice, right, the presence of spines on the dorsal margin. Some Eunotia have spines, some don't, right? And the size and shape and location of them can be particularly helpful. So, something to think about. All right. Linked with that, we have the Rimoportulae. So this is question 3. And again, you can decide how you want to grade this, whether you give yourself full points, partial points, or bonus points. No rules here. Typically, there's a Rimoportulae that is present one per valve, kind of on opposing ends of each respective valve. However, just like everything Eunotia, there's variability, and is definitely something that you want to capture. There can be none or none that have been found or documented. I think sometimes when you're looking under a scanning electron microscope, it can be a challenge to find the Rimoportulae. You know, you can't get the valves to jump under a scanning electron microscope. There can be two, maybe two on one side, or there might be two on opposite ends of each valve, or even within a species. There's an example of a Eunotia population where some valves have one Rimoportulae and some valves have two, one on each end. If you go down into places like Brazil, then you can get more per valve. There's some beautiful Eunotia really showy, charismatic Eunotia hanging out in some of the tropical areas. So you look for that. And on the left-hand side of the screen, where there are some scanning electron micrographs of the Rimoportulae, you can also see that external expression of the Rimoportula pore. And you can think about the location of that and the function of that and how it might help different Eunotia shapes interact with their environment. That's where some of the dance moves are going to come in. Just thinking about the Rimoportula and its location. Now, if you have counted Eunotia before, you know that most of the time you see Eunotia, they are in girdle view. So this is your response to quiz question 4, if I've got it right here. And typically, Eunotia are wider in girdle view than they are in valve view. So I've got some examples here of girdle view and valve view for a variety of different species where you can see that. And you can get a sense of the shape. You can check out patterns of the Raphe in girdle view. You can often see the halictoglossa and kind of where it shows up in girdle view. The combination of those things can help you to figure out what girdle views of Eunotia look like. But my request or plea out there is that if you are going to present a size series of Eunotia valves or valves from Eunotia species, please try and include a few in girdle view. Since that's how we typically see Eunotia, that can be helpful. And I also want you to think about what it means that Eunotia are typically hanging out in girdle view when that's where their Raphe is, and how do they interact with their environment. Right, so this is my slide of Eunotia. Oh, great. Who is here? This is the life. I wish I could see your faces. I'm assuming some of you are nodding, trying to do a count that has Eunotia in it, and it's girdle view 1, girdle view 2, girdle view 3, girdle view 4. Who's there? So again, something that's worth paying attention to. But once you've looked at your slides long enough, you can often figure out what valve view is associated with which girdle view. That can be a good moment. For example, if there are undulations on the bottom left and bottom right of the screen, whether you're capturing the girdle view in kind of the dorsal margin or the ventral margin, you can see those dorsal undulations that are present. So yeah, keep girdle view. And if you're really lucky, you get the one valve where it's in girdle view, and then one of the or frustule that's in girdle view, and you get one valve that's flipped up, and then that increases your confidence. One of the challenges, I think, with looking at Eunotia in girdle view is that the width of the girdle view changes with cell cycle. So there's some nice papers out there where we document that, where it's just after cell division, it's narrowest. Just before cell division, it's widest. That can make it particularly challenging to think about width as a diagnostic characteristic. So with that in mind, there's a paper out there by Miami that was looking for some stable characteristics that didn't vary with cell size. And Shigeki Miami is encouraging people to, if they're describing Eunotia or looking at especially a scanning electron micrograph, and you can get that mantle in view, can you look at the number of striae in the middle of the Raphe on that mantle, kind of above and below? There's some evidence that that's a stable characteristic, and if we started reporting that, we might be able to understand if that's a helpful characteristic or not. But it also, in the process of writing this paper, Shigeki Mayama also was talking about how you measure kind of the width and the width of the valve face, or the mantle area, because you can get that juncture between the valve face and the mantle can be 90 degrees, it can be beveled a little bit, and it can be rounded. So that's something to kind of think about. What is your Eunotia specimen doing? All right. So, thrown a lot out there. I'm going to take a tiny little pause to get you to wrap your brain around some of the things you've just heard. I'd like you to type in the chat area some things that you think you should pay attention to, measure, document when you are observing or describing Eunotia. If you're new to Eunotia, you can type anything. If you are a Eunotia expert, then I want you to go a little bit deeper and type something in that you learned or remembered. So just pause there for a second. And Sylvia, you could turn on your mic and just do a running toss out of things for 30 seconds or so.
Sure, I can do that. There's a comment here from a little bit earlier. Gina quoted a person from the Iowa Diatom class. The road to hell is paved with Eunotia.
What? Any other characteristics on this road that people are paying attention to on their journey to Eunotia heaven?
Yes, we've got hyaline areas in valve view, helictoglossa placement in girdle view, how the frustral looks in girdle view.
Okay.
Mark commented there's an incredible range of valve lengths.
Yes. Yep. I would buy that. Perfect. I can work with those things. Okay. So I'm sure there are more ideas or more that maybe people didn't get typed in. But when you go to the DONA website or any text on Eunotia, and that's one of the challenges of working with Eunotia is a number of the keys are from Europe. There's some really great keys coming out of Europe. For example, Horst Longberlo has a beautiful book on Eunotia, but we need to start understanding what's happening in North America and kind of get more documentation there. So here's an example. I pulled 6 small specimens from the DONA website, right? And you can imagine that, well, when they're all put together, maybe you can tell them apart. But if you had a whole bunch of them, right, what are the different shape features that you might need to pay attention to? These are not to scale. Well, the scale bar's on there, but they're relative to each other. Or here's another set of similar shaped Eunotia species, right? If you put your Eunotia goggle on, are you going to be able to pay attention to the different features and clearly distinguish these different groups? And here's some on the longer end. And if you're familiar with Eunotia, one of the things that you will notice at when you go to the DONA website is we have captured we have some Eunotia pages, but this is my plug out there to people that we need to do some more work with Eunotia, and we need to get more Eunotia pages on the DONA website. So contact me if you want to submit a page, or if you want to collaborate on a page, we can do that. Or if you have a good population and you don't want to have anything to do with Eunotia and you just want to pass it off, if you've got a good population, send it my way, and I can make sure that we do some work to kind of get the DONA website populated a little bit more, so that road to the Eunotia party, that's what it's going to be. The road to the Eunotia party is populated with awesome Eunotia species. so with that in mind, I kind of did a quick search of some of the recent Eunotia species that have been put out there, and many of them globally, there are some kind of hot spots of biodiversity explorations. Southern Brazil, China's got some new Eunotia species coming out. Tropical Central Africa, Vietnam, Spain, Lake Baikal, right? There are, and this is not a comprehensive list, but just some examples of, there's lots of work to be done with Eunotia. And you can see that variation in length and the bottom of the screen, striae density, oofda. Yeah. All right. So if you're thinking about all those variations in length, Rimoportula location, Raphe type and structure, think about the Raphe system, whether you're small or large or wide or skinny or bumps, no bumps, what does that mean if you are out there in the environment? So question 7 was, if you are looking for Eunotia, right, in a diatom scavenger hunt, where would you go? And I suspect that a number of you said, well, we'd go to some sort of acidic environment, right? The strength of Eunotia is they are a pH indicator, right? There are some that can tolerate really low pH levels, right? So really high acidity. They're typically associated with bryophytes. So maybe you're going to the benthos to look at bryophytes in a bog, right? So places that have lots of moss would often be a great place to find Eunotia. But they are pretty much, there's some Eunotia hanging out in lots of different habitats: streams, lakes, bogs, fens, wet walls, chicken feet. I'm just kidding. Yeah, so if you find high numbers of Eunotia in your samples, it can be an indicator that, hey, maybe I need to think about what's going on with the water chemistry and look for those, that type of information. Because they can tolerate really acidic environments, they are often used to track or assess or measure recovery from metal contamination. And this is one of the groups that can show morphological deformities in response to things like heavy metals. And you can actually do a count and you can keep track of at least major morphological deformities. But they can happen internally, externally. But another tool that you can have at your fingertips when working with Eunotia. So typically Eunotia are hanging out in the benthos or associated with bryophytes or on rocks. There are a handful of species that are planktonic, and their morphology also has some cool things. Typically when you think of Eunotia, you think of discoid chloroplasts, but I think there's some variability there. Oftentimes what happens when people are looking at diatoms is they clean the diatoms and they don't ever look at the live specimens. Don't ever look at them, kind of in a gemisch from the environment to see what the growth form of the diatoms are. So I, again, really like to see when people are capturing Eunotia specimens that they capture the live forms, get some live images there. So if that's at all possible, please do that. I also am going to send out a plea there. I've been thinking about, in conjunction with Kalina Manoilov, kind of the characteristics of Eunotia, some of those live characteristics. So if you have some good live images and you want to share and you've got some information associated with them, please send them my way. I would love to start thinking about that a little bit more deeply. You can get them as a single frustule. There are species that form chains. They can be adnate. There are some that spend a little bit more time upright or kind of forms like stellate clusters or even, you know, kind of star-shaped patterns if you're thinking about planktonic forms. Question 8. I think this is so cool. There was a recent description of Eunotia from the Brazilian Amazon area where Eunotia have been documented on stocks. And this is, I think, unusual, and I think is worth some more exploration and thinking about how Eunotia all fit together. But yeah, there's even some kind of that form branched tree-like, or have branched tree-like growth forms. But my point here is, again, I think we don't know a lot about this, and I think we can do a better job of capturing that. So we're coming to the kind of the last part of my presentation here, and my question 9, what does all this variation in form mean for Eunotia? It means that Eunotia got some dance moves. Typically, people don't think about Eunotia as being able to move, but it turns out if you've got a Raphe, right, you can do some movement, right? So they can actually move forward. They can do some pivots, and they can reorient themselves and flip themselves down so they can get back so that their Raphe is in contact with whatever substrate or surface they want to interact with. So I don't know what you had for your responses there, but this is also where I wanted to put a little plug out for a forthcoming volume on motility in diatoms. There's going to be a chapter in there on Eunotia, kind of talking about what we know, what we don't know, but you can look for that. But a little bit of a teaser, and I'll go through this fairly quickly, but people have actually measured movements in Eunotia. And this is where that variation in the Raphe and the Raphe Fisher really come into play, right? Is it recurved? Is it really short? Is it just on the ventral or just on the mantle, right? And when you spend so much time looking at Eunotia in Girdle view, well, turns out that orientation that you see of those Raphe systems in Girdle view allow the Eunotia to kind of move forward in jerky motions, right? That's not smooth like other diatoms, because there are some limitations to that short Raphe system. But they can essentially kind of tip up on one end and move on one set of Raphe endings, you're thinking about in the BC direction there, and then flip over to AD and kind of zigzag along. There's some cool little videos out there. Documented that for a small handful of species, but again, people don't spend a lot of time thinking about what they're seeing under a light microscope. And in long skinny taxa like Eunotia bilunaris, when it's recurved, they can actually do that forward movement a little bit if they're up on their valve end, but I think it's challenging to do. Some of the larger specimens have been documented to if they're laying on their back, they can get themselves flipped over, right, and get their Raphe back in contact with the substrate, right? So kind of it would be the pivot dance move. I'd like to see you do that on the dance floor. You'd be like, let me do the Eunotia. Maybe at our next diatom meeting, some people can do that. And again, depending on what your Raphe structure is, might depend on how you can do that kind of movement. It can pivot or spin, right? You can put anchor one side down, and they can actually use the Raphe on one side and have things happening pulling one way and pulling the other to allow it to kind of do a spin. So some cool things there. Yeah. And there's if you have an understanding of physics, which I don't have a great understanding of, but that the location of the external expression of the Rimoportulae pore, right, might be an anchoring point that would allow you to then move along the Raphe and get the valve or frustule flipped over. This wouldn't be a valve. So we've done a tour of the Eunotia. We've talked about the characteristics. We've talked about the variation in form. structure and thinking about it in terms of the environment and thinking a little tiny little bit about movement. Lots of room for more work, lots of cool things to observe, lots of potential and opportunity here for collaboration. So don't be shy about contacting me if you want to work on donor websites, or if you've got questions about Eunotia, cool images, live images, or you've got something you want to do together. So I think I will end here, ask you how you did on your quiz, and take any questions.
Thank you very much, Paula. That was fascinating. We've got some answers here for the dance moves. We've got twist and waltz, shimmy, another twist, and a twerk. I thought because of their small Raphe, they might move in a slow circle, slow dance.
Oh, I love it. Yeah, they're probably a little bit lower side, but there's Harbeck in, I can't remember where he's at. He is a physicist, but he's got a whole page on Eunotia and some movement there. So when the chapter comes out, you can get the links to that. Go check it out.
Will do. So anyone who has questions, please feel free to put that in the chat. There was one comment above where Anna talked about a Striae in the middle of the Raphe, and maybe I missed that. Do you know what she might be referring to?
My guess is when I was talking about the Raphe, when you're looking at the Raphe on the mantle, and there can be striae above and below the Raphe, just as a part of the mantle, paying attention to what those I guess what the striae on the mantle are doing there. Mm-hmm. And how many areolae are, if you took that Raphe in Girdle view and you were, I guess you'd have to probably do it under the SEM, midway on the Raphe, how many areolae are above and how many are below? There's some evidence that that's a stable characteristic. I don't know. I don't know that we've explored that enough. That's my guess what the question was.
Okay. We've got more questions here. Seth asks, can you talk about the variability in Raphe morphology within a species versus between species?
So I think in a general sense, within a species, the Raphe characteristics are a little bit more similar. So for example, if it's strongly recurved, it typically strongly recurved within a species, you would find that recurve nature in most of the valves in the species. The things that I have seen within a species in terms of variability of the Raphe might be if it's extending up onto the valve face and there's maybe like a little bit of a C, that sometimes that isn't a smooth C. Sometimes you'll see like it'll do a little zig or a little zag or maybe flip up, right? So there can be subtler variation kind of in the layout of that. And I don't know whether that would be considered a deformity or whether that's something that species does where there's a little bit of subtle variation in shape. Those are things that I have seen.
Seth gives a thumbs up.
Thanks, Seth.
Mark asks, do you think that the hymens are variable in their presence among Eunotia or simply lost with cleaning of samples, those occlusions on the areolae?
That's a good question. I mean, we all clean the diatoms pretty aggressively, and I think there's been some advancements in scanning electron scanning electron microscope capabilities. But when people who take really high resolution, high quality scanning electron microscope images show really clean areolae and really like really delineated hyaline areas. So I think my sense is that it's a characteristic. I'd love to hear what maybe was prompting that question, or if you've seen some observations there. Is that Mark?
Yeah.
I was only thinking that I find it variable in Eunotia. And several times when I've worked on Eunotia, I've seen hyaline, and other times the material ends up not having them. And I just wonder if they get blown out when you clean your samples or store your samples for long periods of time. So that was the only reason I was asking, just what your
yeah, thank you for sharing that. I don't have a good sense of that, other than kind of people who pay attention to that or pay attention to how they clean it, and have really high resolution SEM images have shown some things. But that might be, you know, an interesting thing to explore a little bit more. How does that play out in Eunotia? So Mark has dropped a gauntlet there, people.
We have a question from Kalina. She says, great talk. Her question is, will Eunotia make spores, especially the ones that make chains?
Yes. I have some good images of spores in Eunotia, and the chain forming ones. And you kind of see nice round spore showing up, at least what I would call a spore. Yeah.
I have a question which was prompted by Mark's earlier comment about the really huge range and size within a species, and was wondering if you know, it might be due to the environment that they live in, that they will, you know, keep dividing until they're really small, and not, you know, go through that size regeneration with sexual reproduction very, very much.
You know, I think they're like any other diatom that's out there, right? That there seems to be a general upper and lower limit. But in saying that, there are some species that have a really huge range. And then there are other species where the range would be more what I would call reasonable. The Eunotia would be unreasonable. No. Yeah, I just think there's lots of variability. Some species have that huge range. And that's where, you know, doing like you would do with any diatom description or whatever, you capture the smaller valves and the longer valves. And if you can capture an initial valve, that can be really helpful. And then when they get smaller and smaller and smaller, they almost end up like little round nubs. You know, and then you're kind of like, okay, it's not going to get much smaller. I don't know if that answers your question or not, but it's very.
Yeah. I also had a question about, you know, you talked in the beginning part that there's a lot of variation in morphology that is not necessarily due to their size. So in those kind of taxa, what would be your advice about defining species morphological boundaries?
I think that it is one of the challenges of describing Eunotia. And so, for example, like on the DONA website, I think what we need to do is have kind of the typical species morphology, and then have, you know, like if you were making a voucher flora or something, show some of that morphological variation within a species, and just see if we can start to wrap our brain around it. You know, like, is that variation happening in response to a particular environmental cue? But it really does make it challenging, back to whoever's quote and the road, counting Eunotia, if you've got some similar shaped species in the same population and there's some overlap and you're trying to use valve length or valve width, or, you know, is this end fat or thin? I think it's why, you know, people pull their hair out and you end up with like, maybe, well, I'm just going to make this a species cluster or complex that I've got a couple different species in here, but I can't consistently tell them apart. Maybe we need to do some genetic or get some genomes of these species, and maybe there's cryptic diversity, and maybe what I'm saying is variation within a species is really something else going on. Who knows?
That would not surprise me.
Yeah, but it does make it challenging. Yeah.
Yeah. All right, we got one last question and we'll wrap up. Anna says she's read that Eunotia cells are sometimes found inside sphagnum mosses, and she wondered, how do they get in there?
I have no clue. You're talking about like inside a cell or inside the like the where the little leaflets or whatever?
I am not sure.
Because in the little grooves, like if you've got a little microhabitat there, they will totally hang out there. And some of the movement pieces that I was talking about, where like on a slide you could get Eunotia attached to the cover slip and then drop down to the slide and orient itself. I could see some of those things that people have observed under a light microscope being really valuable moves to be able to do in a small microhabitat, where you could take advantage of all surfaces, you know, in terms of food acquisition, you know, kind of getting resources or moving away from predators, or just orienting yourself so that your Raphe is on the surface. So yeah, more work to be done.
Yeah, wonderful. Well, we are past the top of the hour. I want to thank Paula again for making us more familiar with Eunotia and the Eunotia party.
Yeah.
And
We're going to change that phrase. Very important.
And we will see you all again in a couple weeks for a talk by Vicki Striae and Shelley Wu on diatom education. Thanks, everyone.
Thank you all.
Thanks, Paula.